Okay, so I'm drinking a shake while I talk to you. You guys know my shakes. I bring them to class all the time. We have one more set of research findings that I want to share with you before we make a conclusion about this whole sex addiction enterprise. Is this a real thing? Does it make sense? Does it make sense to draw parallels between sex addiction and drug and alcohol addiction or not? Let me talk about one last set of findings involving compulsive sexual behavior. What does the research say? The largest volume of studies by far that is associated with compulsive sexual behavior indicates a neurobiological basis for compulsive sexual behavior similar to drug and alcohol addiction with regard to a specific variant of a gene. So I'm going to get a little technical here. Time out for technical. Again, I'm aware that we have a variety of folks, a variety of majors and minors in this class. Some of you are very well versed in genetics and biochemistry. Some of you have less experience with this. We'll do my best to explain things to you at the level necessary to understand the research. Transcription factor delta phos B. Delta phos B. And interestingly enough, this is associated with the area of the brain most critical to drug and alcohol addiction. You guessed it, the nucleus accumbens. By the way, compulsive sexual behavior made my dog Silas here decide it was time to hump some blankets. So right on cue, here he goes. Sorry about that, but he has a mind of his own. I think he understands English sometimes. I really, really do. Anyway, back to delta phos B. You may remember from a prior discussion when we talked about drugs and alcohol addiction. We have gene alleles. And remember what this means, gene alleles. We have alternate forms of genes on the same chromosomes that are passed down genetically to different people. And these alternate forms are associated with different behavioral patterns, different phenotypic traits. Remember what phenotype means. Genotype is your actual genetic material. Phenotype are the observable characteristics that we see that are influenced in part by our genetic material. So here we have a situation where delta phos B is an alternate form, a variant of the phos gene family. It's an allele. And it is a transcription factor. So what does that fancy word mean? It means a protein. It's a protein. It's a transcription factor is a protein that binds to DNA, to your genetic material, and regulates how that gene is actually expressed. So phos B is encoded by the phos B gene on the human chromosome 19. Researchers actually know the chromosome that these alleles, these alternate forms of this gene, are coded on. Now, in the nucleus accumbens, that very important area of your limbic system that has come up time and time and time again with regard to research on drug and alcohol addiction, in the nucleus accumbens, the delta phos B transcription factor functions as a sort of molecular switch, or a master control protein, as it's been called. And in the development of addiction, it can impact addictive processes if it is turned on. So without getting too complicated, there is an entire field of study known as epigenetics. Epigenetics. And what's really fascinating about epigenetics is it explains why people with potentially the same genetic structure can have different phenotypic traits. Because the way your genes are expressed is critical for your phenotype. And whether certain genes you've inherited get expressed or remain dormant depends on whether they are turned on or left off. Now, this delta phos B in the nucleus accumbens, if it is turned on, or in other words, if it is overexpressed, then it triggers a series of events that will ultimately, researchers indicate, produce an addictive state, compulsive reward seeking involving whatever the target stimulus is that the person has become overly involved in seeking out and engaging with. Okay? So what the research indicates is that just like drug and alcohol addiction can involve overexpression of this delta phosphB in the nucleus accumbens and therefore compulsive drug and alcohol seeking, potentially delta phosphB can also become overly expressed in the nucleus accumbens for people who are involved in compulsive sexual activity. And much like with alcohol and drugs, people become very focused on the reward associated with drug and alcohol use and seeking drugs, the pleasure, and also the importance of it that is signaled by that brain area being overstimulated again and again and again. And just like people with drug and alcohol can become overly sensitized to any cues related to their drug and alcohol use because of classical conditioning, and it makes the brain start to react the moment they see that cue or in some way are presented with that cue. Just like people with drug and alcohol addiction have heightened vulnerability to relapse and involvement in those compulsive behavioral drug seeking after some remission, the argument is these same things can happen with compulsive sexual behavior because of delta phosphB. Now, I will say this line of research has one limitation, and that is it really has to utilize non-human animals, generally speaking. Mice, rats, hamsters. Many people argue that is not the same as a brain of a human. But we have to use mice, rats, and hamsters because a requirement of this kind of research is euthanizing the subject of the research in order to access and measure intracranial delta phosphB post-mortem. You can't, in general, guys, I should tell you this if you don't already know it from your perhaps biological types of classes you've taken. We can't really get an accurate measure of things like neurotransmitters or other chemicals in the brain in a live subject. There's too much metabolic activity going on at any given point in time to get an accurate measure because your chemicals aren't static when you're alive. Depending on what you're doing, depending on brain activity, things are being metabolized at different points in time. So you really need a post-mortem examination. Silas, stop! You really need a post-mortem examination of a brain to get a sense of how much of a particular chemical in the brain has accumulated. So to really, Silas, stop! Sorry, guys. To get a sense of how much delta phosphB has been expressed in a brain area, in this case in the nucleus accumbens, you need to euthanize, or in other words, unfortunately kill, end the life of the mice, the rats, the hamster, and then measure it after death. Multiple tests using hamsters who were purposely bred. And this is the other piece of it. You can control conditions with these animals because you can purposely breed animals to express a particular allele or form of the gene, compare them to animals not bred to express that allele or form of the gene, and see how that changes their behavior. In this case, multiple studies using hamsters who were purposefully bred to overproduce delta phosphB have focused on the impact of this on that animal's sexual behavior, and they found enhanced sensitivity to sexual activity in the animals who were purposefully bred to overproduce delta phosphB. In 2008, researchers Wallace and colleagues naturally induced this sensitivity in laboratory rats via, quote, chronic sexual behavior. So they had these animals engaging in repeat sexual behavior to see how it changed their brains. What the researchers found is that repeat sexual experience significantly increased the concentration of delta phosphB levels in the nucleus accumbens. Interestingly enough, compared with control rats who were not made to engage in excessive sexual behavior. Although the rates of increase in terms of delta phosphB concentration in the nucleus accumbens in these animals was lower on average than when the rats were tested for excessive drug activity, and you know that research from prior classes, we can get animals to engage in compulsive drug seeking. Although the delta phosphB accumulation in the nucleus accumbens on average was lower for rats with compulsive sexual behavior, induced to engage in compulsive sexual behavior, than rats induced to engage in compulsive drug use. Nonetheless, compared to control rats, both groups of animals had higher concentrations of delta-phosphate in the nucleus accumbens, whether we're talking about rats who were made to engage in compulsive sexual activity or rats who were made to engage in compulsive drug use. 2010, researchers by the name of Pilchers and colleagues illustrated the production of high levels of delta-phosphate, again in the nucleus accumbens of mice, is correlated with stronger reinforcing effects of sexual reward. So again, the more they were engaging in sexual activity, these laboratory rats, the greater, laboratory mice rather in this case, the 2010 study was mice, the more they were engaging in sexual behavior, the greater the accumulation of delta-phosphate in the nucleus accumbens of the mice as measured post-mortem in their brains. Also, these same researchers, Pilchers and colleagues, found mice have increased sensitivity to amphetamine drugs. These are central nervous system stimulants after repeated sexual experiences. So what the researchers took away from this is the very same animals, when given high levels of amphetamines, are also made to engage in high levels of sexual activity. These very same animals show similar increases in delta-phosphate, this important transcription factor, in the same area of the brain, the nucleus accumbens, which we know is critical in out-of-control compulsive drug-seeking and alcoholic behavior. And so they argue these brain parallels cannot be ignored. That it's interesting that the very same brain area and the very same gene allele and the very same overexpression of that gene allele is associated with both compulsive drug and alcohol seeking and compulsive sexual behavior. They say there has to be a parallel here. There has to be parallels in the underlying changes that happen in the brain with either form of compulsive behavior, be it drugs and alcohol or be it sexual behavior. So I would really like you, before you go on to the next video for slide number 10, I would really like you to take some time to reflect on the research from slides 6, 7, 8, and 9 that I have reviewed with you here that involve the brain in some form, that involve animals, that involve humans and pornographic use. And I want you, before you go on to slide 10, I want you to kind of form your own opinion. What are your thoughts given this body of research, albeit only over a 3-year recent period, 2014, 15, 16? What conclusion do you come to? Do you think this research is sufficient to argue that, quote, sex addiction is a bona fide condition, that it is influenced by the same reward pathways in the brain, that the same reward pathways are hijacked, that the same underlying genetic and biochemical components underlie both of these behaviors, drug and alcohol and compulsive sex? Or do you feel like the story is still out and it's too premature to say that sex addiction is a bona fide disorder that should be diagnosable and treated and has the same underlying biochemical influences? Take some time to come to that conclusion yourself before you move on to slide number 10, and then I will see you on slide number 10.